I'm going to give you an example today, a classic example of adaptation, and that is the ability to digest milk as an adult, referred to commonly as lactose tolerance. And we are going to compare, for example, on the left is an African population that has practices cattle herding. We refer to them as pastoralists. And on the right, we have a European farmer who practices dairying. So milk contains very high quantities of the sugar lactose, and the molecular composition is shown on the milk bottle here. Now, in order to digest the sugar lactose, you need expression of an enzyme called lactase. And lactase is going to chop it up, that lactose, into its component sugars, glucose and galactose. And if you're able to digest the milk, and if you have the active enzyme, that's going to be rapidly taken up into your bloodstream. And I also want to mention that the lactase enzyme is expressed specifically in your small intestine. Now, almost every baby is able to drink milk, right? So everybody's got an active form of this enzyme. However, shortly after weaning, in all mammals and in many human populations, this enzyme is shut off. Okay, so that means that as an adult, you don't have an active form of the enzyme, and you cannot digest the sugar lactose that's present in milk. So often that's referred to as lactose intolerance. But in some people, due to a genetic mutation that I'm going to tell you more about, they maintain the expression of this gene at high level as adults, and they can digest the sugar lactose, and we say that they are lactose tolerant. We can also say that because the enzyme is maintained as active, we could say they're also lactase persistent. They have the lactase persistence trait.
In eukaryotes, DNA transcription begins when general transcription factors bind to the promoter region of the gene. Those transcription factors facilitate the binding of the RNA polymerase enzyme to the start position of the gene. RNA polymerase needs to be activated by a type of transcription factor called activator. Some activators recognize and bind to the regulatory region of the DNA near the promoter region. Other activators bind to a distant region of the chromosome called the enhancer region. Activators bound to the enhancer region work with mediator molecules to initiate transcription. Another type of transcription factor, called repressor, inhibits the activation of transcription. Repressors can act in different ways. Some repressors bind to the binding site of activators and prevent activators from binding to DNA. Some interfere with the molecular interaction between the activators and the RNA polymerase. Some activators and repressors act by affecting chromatin structure. These repressors can alter the chromatin structure to make it coil up tightly. Coiled up genes are not available for transcription. Conversely, activators can uncoil a coiled section of DNA and make genes in those regions available for transcription. The interactions of all these factors determine the actual rate of DNA transcription.
quite some time, anthropologists have noted this very interesting distribution. In sort of the brighter colored purple here, that is showing the prevalence of the lactase persistence or lactose tolerance trait around the world. And it's long been known that this trait is at highest prevalence in Northern Europe. So in the Finnish population, something like 99% of the population is able to digest milk. As we move into Europe, into Southern Europe, and then into the Middle East, we see less and less and less of that trait. As we move into East Asia, very rare. The Americas, very rare. Africa, very rare. So one take-home point you should remember is that the ability to digest milk is a recently derived trait in populations whose ancestors practice dairying and they drink milk. And most of the world cannot digest milk. Keep that in mind. If we look at the lactase gene, it's a really big gene. It's like 50,000 nucleotides, and it's on chromosome 2. It's shown in yellow here. Not in the gene, but near the gene, upstream of the gene, about 14,000 nucleotides away is where we and others have found the mutations that regulate the expression of this gene. In green is shown the mutation that regulates this trait in Europeans. So people who have a T at that particular position can digest lactose in milk, and those who have a C cannot. You have 2 copies of the C. In Africa, we found a different variant. It was about 100 nucleotides away from the European variant. And if you have a C, you could digest milk, and if you have 2 copies of the G wild type, you cannot. And what it appears is that these 2 mutations occur in a regulatory region, a region that's going to be important for regulating gene expression. We call that an enhancer.
Okay.